All right, folks, I'm going to do another lecture on viruses. Unfortunately, I had one that I did yesterday. In the middle of it, I guess my internet cut out. There you go. There's a problem that you can have doing this kind of thing. So anyway, I want to get back into the viruses, and I'll do another lecture, perhaps two, however many it takes to get through this. So I'm going to go ahead and get us back into the PowerPoint that I was looking at yesterday. Um, I think we got through all of this business here yesterday with viruses. The general information. That they're very small as microbes go. That they're pretty simple. They're composed of capsids and genetic material, and they may or may not have envelopes. They may or may not have matrix proteins and enzymes. Went through the different capsid types. Icosahedral capsids look like this. They're composed of little triangular plates of protein that are wedged together to form this interesting sort of organized looking geometrical shape. 20 sides and 12 corners, like that. And then we've got forms that are helical, where the capsimers are rolled into a little spiral tube, and the genetic material is contained in there. We have forms that are naked that have icosahedral capsids that have little spikes on them like this. We have forms that are enveloped that have this covering composed of phospholipid bilayer derived from the host cell, and they likewise can have these little spikes composed of proteins of different types that are used for attachment and entry into a host cell. All right. We talked about, or I've covered a little bit of capsid and envelope structure and functions. We've gone through all of that. Talked about the fact that there are some complex viruses that have a mixture of characteristics, so they're very difficult to classify. These phages, or bacteriophages as they're called, are very, very common viruses out there in the environment because they occur wherever there are bacteria. And I'll just make this point. Every organism on the planet is subject to viral infection. So you name an organism and there will be a virus or viruses that infect it. Because bacteria are so ubiquitous, it stands to reason that these bacteriophages are going to be ubiquitous as well. So this is a bacteriophage here, and you can see at the top it's got this naked icosahedral capsid with the genetic material, and generally that's going to be a DNA. There's different forms that it takes. It can be linear, it can be circular, it can be double-stranded, it can be single-stranded. There are a few RNA viruses that are bacteriophages. Anyway, it's got the little icosahedral capsid, it's got this helical sheath, and it's got these little leg-like tail fibers, and they adhere to a host cell, and this all plunges down here and literally shoots the genetic material into the host cell. So here this kind of shows all of the different variations that you might expect to see among the viruses. I did mention earlier the complex viruses also include the pox viruses, which are DNA viruses that animals can be infected with. Smallpox, cowpox, those kinds of things were diseases that historically affected humans in a really, really serious way. They have these outer coverings that are unusual in that they're composed of lipoprotein fibers just wrapped continuously around the virus. That's unusual. That's not something we typically see. Anyway, this is a micrograph of a helical naked virus. All right, so now we want to get into discussion of nucleic acids. So go ahead and expand this. Yeah, the genetic material of a virus can be either DNA or RNA. You're not going to have both of those things present in a viral particle at the same time. It's a valuable thing to recognize because you can classify viruses on the basis of whether they're DNA or RNA viruses. And what's more, there's variation among viruses in terms of the type of DNA or the type of RNA that they have. That's something that's unusual because living things, all of the living domains of organisms, have double-stranded DNA as their genetic material. That's everything, prokaryotes and eukaryotes alike. But in the viruses, there are some forms that have double-stranded DNA, but there's some that have single-stranded DNA. And likewise, there are some forms that have RNA as their genetic material, and it can be double-stranded or single-stranded. And there's even more variation beyond that, how the genome is structured. In some cases, forms have circular genetic material. The DNA or the RNA is in a circle, and others they're straight, linear pieces that have two ends. Some of them have one piece of genetic material. Some of them have multiple pieces of genetic material. So there's a lot of variety among the viruses in terms of those things. But in all cases, the genetic material contains genes, segments of those molecules that code for proteins, and those proteins in turn become either structural or they are enzymatic, and they're involved in helping to form the virus so that they continue to increase in number and move from host to host and persist that way. So the genes that they have control their traits, just as is the case with living things. The number of genes that are needed is typically pretty small. The genome size is from, say 8 or 9 genes total to maybe up to 100 or so, a few hundred. That compares with larger, complex eukaryotic organisms whose genomes number in the thousands, 20,000 genes in humans, somewhere in that ballpark would be typical. So simpler things require smaller genomes, more complicated things require bigger genomes. It stands to reason that that's going to be the case. All right, so among the DNA viruses, like I said, we have forms that are double-stranded, and they're usually designated with this lowercase ds. All right, so if you see a virus's description and somewhere written there is that designation, dsDNA, that tells us that it has double-stranded DNA. There are a bunch of different viruses, some of which are pretty well known, that are double-stranded DNA viruses. The human papillomaviruses, for example, herpes viruses. And then there are some forms that have single-stranded DNA, so that would be designated simply with a pair of lowercase s's and DNA. So that would tell us those are single-stranded, and there's a bunch of those. And like I said earlier, they can either be genomes that are circular, as is the case in prokaryotes. They have circular pieces of DNA, and then sometimes they have linear pieces with two ends. All right, so just it varies from group to group among the viruses. And then the RNA viruses may either be single-stranded or double-stranded as well. There is this phenomenon of segmentation that we see in some of the RNA viruses. That's where they have multiple pieces of RNA with genes on each piece. The interesting thing about those viruses, and there's not a whole bunch of those, but there's a few. Interesting thing about those is that they can actually swap pieces of RNA with other viruses. So if you have a cell that's infected with 2 or more viruses, and that can happen, if it's a virus that has segmented genetic material and it happens to be in there with another virus that has uh, even just a single piece of RNA for its genome, they can actually swap pieces of RNA and and acquire new genetic traits as a result of that. So that's something that would facilitate the evolution of some of the viruses. So there are some that have segmented genomes, and they evolve typically very rapidly. In fact, the RNA viruses generally evolve faster than the DNA viruses because in copying RNA, there are no proofreading mechanisms. And what that means is if there's a mutation or actually a mistake in copying the RNA to make new viruses, that mistake is not corrected. And sometimes those mistakes are bad for the virus, they'll destroy it. Sometimes those mistakes cause changes that are, adaptive or beneficial to the virus. So, and the mistake rate, the mutation rate in RNA viruses is sometimes hundreds of thousands, if not millions of times greater than in other types of viruses, the DNA viruses. So they evolve rapidly. And some of the viruses that are problematic for us that, infect humans that we try to deal with using vaccination evolve so rapidly that we have to, periodically come up with new vaccines to deal with that. All right. So influenza viruses, you might guess from knowing that we have to come up with a new vaccine for influenza viruses basically on an annual basis, that they are RNA viruses. And you would be correct. They are RNA viruses. The coronavirus that we're dealing with now, which is one of the SARS viruses, SARS coronavirus 2, is the virus that causes the disease COVID-19. It likewise is a single-stranded RNA virus. So they evolve rapidly. And the RNA viruses can fall into 2 categories. There are those that are positive sense, which means that once that RNA enters a host cell, it can be used immediately and essentially works as a messenger RNA, which codes right away for protein. So it doesn't have to go through any major processing to be utilized. It uses the host cell's ribosomes and transfer RNAs and goes about then hijacking that system so that it can make its own protein parts for capsids and enzymes and so on. So, and the coronavirus we're dealing with now is a positive sense RNA virus. Then there are negative sense RNA viruses. So what they do is they utilize enzymes that are present in a cell. They inject their negative sense RNA. They use that as a template for making positive sense RNA, which then can go right away into use as the information to make proteins, and goes from there. So the positive sense forms just go right into the cell, and boom, proteins are produced using those. All right. And by the way, anytime RNA is used to make protein, if this is happening in your cells, it would be the same thing. We call that translation. Translation is making protein using the information in RNA. Transcription is where we go from DNA to RNA. Something everybody hopefully members from ANP1 or past somewhere. So let me just write that down, just so you remember. I've talked about it before. Transcription is where you go from DNA to RNA. messenger RNA in our case, which is the same as positive sense RNA in a virus. And then the step where you go from RNA to protein is translation. And I apologize again for the sloppiness of this. It's just really a challenge for me to write with my finger. That's what I'm doing. That's protein. Anyway, so this discussion of nucleic acids and viruses. Put the dog out. Go out. Go. No, don't. This discussion of nucleic acids and viruses takes us to the discussion of classification of viruses, because as it turns out, the genetic material of the virus is one of the main aspects of the virus that helps us in classifying. And there's some other things that go into this, but there was a classification system developed by a famous virologist whose name happened to be Baltimore, David Baltimore, and it is the classification that is basically used by all virologists now because it works. And so there are 7 different groups of viruses in this classification. These Roman numerals 1 through 7 over here correspond to the different groups of viruses that are recognized. And you can see the first 2 groups are DNA viruses. Group 1 is double-stranded DNA. And the DNA is transcribed into messenger RNA. Then that's used to make protein. So that's kind of what happens. This is sort of the standard thing here. We expect to see in organisms, living things. So these are viruses whose genetic material is used in the same way that cellular organisms would use it. So herpes viruses, that family of viruses are like that. And then we have the single-stranded DNA viruses. They convert their single-stranded DNA to a double-stranded DNA, and then that is transcribed into RNA. So there's a little bit of a processing step initially. So the parvoviruses are like that. And then the remaining viruses have some different stuff going on. The groups 3 through 6 are all RNA viruses. But you can see lots of variation in them, like group 3 is double-stranded RNA, which they use to produce a messenger RNA. Just use the RNA as a pattern for making more RNA, and there they can make protein. Pretty straightforward. This group here, the group 4 viruses, the single-stranded RNA viruses that are positive sense, the coronaviruses are like that, the picornaviruses, which include those that cause common cold, and others are in that particular group. Their genomes are used right away to make protein. And then we have the group 5 forms that have negative sense RNA. Their RNA has to be turned into a positive sense RNA before it can be utilized. Rabies viruses are like that. And then down here, group 6 is interesting. These are the so-called retroviruses, right? Retroviruses. So they have an RNA, a single-stranded RNA genome. And what happens with that is it is used to reverse transcribe and form a DNA. And so they use this enzyme called reverse transcriptase, okay? And that's unique to them. There's no other virus that has that. So the retroviruses are basically going in reverse initially. Their RNA is used to make some DNA. And before that DNA can be utilized, it has to go into the nucleus of the host. And there it's incorporated into the DNA of the host. All right, so this was in a human cell. One of the chromosomes, one of the 23 pairs of chromosomes that occur in that cell would suddenly be changed a little bit because it would be uh, melded with or joined up with, would have inserted into it a piece of viral DNA. All right. So this actually gets incorporated into one of your chromosomes. And then, what'll happen sometimes is it just sits in that chromosome, and every time that cell divides, you make a copy of the retroviral genome, right? So if that cell divides, I don't know, 100 times, 1,000 times, 1,000,000 times, every time there's a division, you're going to make a copy of that virus. But it does not cause symptoms usually while it's in that state. So it's sort of latent. It's sort of just laying there. Later on, it can then begin to code for, um, messenger RNA, which then is translated into protein, and then you start to see symptoms. So often that's the case with these particular types of viruses. And so HIV is one of those types of viruses. It is a retrovirus that causes slow infection usually. It takes a long time for the symptoms to arise and for the outcome to cases become pretty dire for the host. Anyway, those are the retroviruses, and then we have the double-stranded DNA viruses that likewise have a reverse transcriptase. So that DNA is used to produce an RNA intermediate, and then that can be used as a template to make messenger RNA, which can be used as the code for protein or proteins that would be used in making capsid materials and so forth, or it could be used to make more messenger RNAs. So anyway, that's an unusual group, and the hepatitis B virus, among others, are in that group. And you can see in the name here for that group, they're the hepadnaviruses. You see HEPA in there, that refers to liver. So these affect liver cells, obviously. And there's DNA, so HEPA DNA viruses. And that tells us they are DNA viruses. Just as is the case here, I just noted there's a misspelling in this where these are the picornaviruses. It should be PIC, not PIR, picornavirus. So PICO, PICO means little. And then you got RNA. So these are viruses that have little short pieces of RNA for their genome. Some of these names are not as technical as you might guess. Anyway, so there's that. There's this Baltimore classification. I'm not going to ask a bunch of really difficult questions about that, but you should know that this classification exists and that it's mainly founded on the type of genetic material that the virus has. And here's a more complete, sort of comprehensive picture that shows the array of different RNA and DNA virus types. And down here you can see little pictures of them that show some of them being naked, and then you see the others that are enveloped. And, lots of variation among them. And it tells what type of capsid they have, if it's helical, if it's icosahedral. See these terms in here, DS and SS, you now know what those mean. Those are the type of genetic material they have, whether it's double or single-stranded. You can see here which ones have segmented genomes, like this guy here. Those Virna viruses, the reoviruses, they're segmented. The Bunya, the Orthomyxo viruses are segmented. Lots of variation. Some of these you probably have never heard of, but these are infecting all organisms. Virtually all organisms can be infected by viruses. Those are the known groups. I'm certain there are others that are out there that have yet to be detected. That's that's a challenge. These things are tough to, find in the environment. We can go out and just sample water or soil or different environmental substrates and look for DNAs and RNAs in there that are clearly not from truly living things, um, and by virtue of that, we can sometimes find viruses, but that's a big challenge. All right, so another thing we expect to see besides genetic material in the core of the virus, we may find preformed enzymes. Some viruses, when they're being assembled, will be equipped with these. And so they're going to be used once that virus finds a host cell. In order for the virus to get rolling, for its infection to take off, it may need a polymerase. Um, so, that could be used to take a DNA template and use it to make some RNA. It could be used to take an RNA template and DNA from it. So there's those transcriptases and so forth. Those are types of polymerases. And then we have replicases. So if you've got a piece of RNA and you want to make an exact copy of that same piece of RNA, you can use a replicase. That's what those are used for replicating multiple copies of genomes that are going to go into however many viral particles that are going to be produced. And then there are the reverse transcriptases, which are used to make RNA using a DNA template, or make DNA using an RNA template to go in reverse. The AIDS viruses, the HIV virus is one of the forms that do that, the retrovirus. So now in any viral multiplication, there are it's like a cycle that has multiple steps. And for the virus to be successful, so that it can spread to new hosts, which is kind of the name of the game. It's got to spread to new hosts and keep doing that, otherwise the virus would go extinct. All of these steps would have to be accomplished successfully in order for that to happen. And so this is useful information because if you say, for example, we're charged with designing a drug, an antiviral drug, you can look at all of these different steps here and maybe come up with a drug that would inhibit one or more of them. So find the weak links, if you will, in the process. If you've got a drug that could, say, prevent adsorption, which is where the virus attaches to the host cell, you give a patient something like that that would block that, then viral infection could be prevented or at least limited. Penetration is where the genome of the virus enters. If you could block that or any of these other steps, then you could successfully maybe treat a virus. So currently, hopefully, that's what they're working on with the coronavirus that we're dealing with. But anyway, in animal virus multiplication, you've got all of these steps. And as we'll see, when we look at other types of viruses, in particular the bacteriophages, there's some subtle differences between what happens there and what we see in animal viruses. Animal cells, you'll recall, don't have cell walls. They never have a cell wall. Remember, that's a rigid sort of barrier that lies outside of the phospholipid bilayer. So the animal cell has glycocalyx of glycoproteins, things, and then you go from that to this sort of fragile delicate cell membrane, the phospholipid bilayer. So if an organism has a cell wall, that complicates things a little bit. It's harder to get through that. And so the multiplication cycle for those particular viruses that are targeting the cell wall possessing forms, and their multiplication cycles are going to be. Anyway, these are the steps that are typical in an animal type of virus, something that would infect animals. adsorption is basically binding to the host cell, using often spikes that are found in an envelope or on the outer portion of the capsid. Those molecules have sort of a geometric fit with those that are found in receptors, say on the host cell membrane, the lock and key fit. That's going to determine what hosts the virus can infect. And it may actually be so specific that it determines what cells in the host. If it's a multicellular organism with tissues and organs, there may be just certain tissues, certain organs that can be infected. Not all of those cells are going to have exactly the same receptors. The coronavirus we're dealing with now infects preferentially cells in the respiratory system. Right? Those cells that, say, are found in the epithelium of the trachea and the bronchi, in the lungs, are equipped with receptors that this coronavirus recognizes and attaches, and that's how it gets in. So if the coronavirus were to attach to, say, or try to attach to cells on your skin or in your liver or in your reproductive tract or wherever, it can't happen because those cells don't have the receptors for it. It's just in our respiratory system, right? Those mucous membranes. And that's why we're saying wash your hands and avoid touching your face so that you don't introduce those viruses to those sites where it can gain entry, gain access to those tissues. But once it adsorbs, then it has to penetrate. And the main thing is getting the genome in there, right? The whole virus doesn't have to go in, although that frequently happens. The key is the genome has to get in somehow, some way, right? Once that happens, once the RNA or the DNA gets in, if it comes in wrapped in a capsid, it's got to be uncoded, right? And so there's that step. And then once it's uncoded, the genetic material can be operative and can be used to code for the parts of the virus. So capsimer is used to make the capsid. The enzymes that the virus would need for all of the different activities it might engage in, those have to be made. And so there's the synthesis. The spikes that are found in the envelope or on the capsid somewhere, those have to be synthesized. So everything the virus needs to become competent as an infectious agent, that would be those things would be synthesized. Then they're put together. So if you could actually watch a host cell that's been infected, if you could see the time sequence of things going forward, you would see ultimately new viral particles being assembled inside of it. That's assembly. And then finally, once the viruses are formed, they have to be released. Otherwise, that it would be pointless to assemble viruses and not have them go forward to infect the cell. So there's this release, and there's different ways that happens. It can happen through budding, or it can happen through lysis. So we'll look at those things, how they're different. So here is an typical animal virus. This particular one has enveloped characteristic. It's got an envelope on the outside with spikes embedded in that. The capsid in here is icosahedral. You can see the genetic material inside of it. So this particular virus would attach to receptors on the host cell membrane. So that's what's going on right there. So there's adsorption. All right. There's a good fit between the spikes and the receptors. This thing hangs on, and that gets it in position where this next step can happen. So this is one possible way that the genome can come in, and this involves, in this case, I'll write this out here, that's endocytosis that you're seeing. My time looks okay, I guess. Endocytosis And this involves invagination of the host cell's membrane. That sometimes just the binding of the receptors and the spikes more or less stimulate that process to happen. Once, the cell has this thing bind to it, it just responds automatically that way. Although it's not advantageous for the cell to do that, it doesn't know any better. They don't have a brain. I think it's something that that they should be taking up. Like some food molecule. But anyway, they take it in, and so obviously this virus now is going to be wrapped in an envelope plus some more of a covering. It's a vesicle or vacuole essentially, because it's got an extra wrapper of host cell membrane around it. Of course, that has to eventually be eliminated, which is what's happening here in step 3. The whole covering, the 2 layers of phospholipid, and the capsid itself are taken apart, are torn down, and that releases the genetic material. So once that happens, if there has to be a reverse transcription step, that's done. If there has to be the formation of a positive sense RNA, that's done. Whatever it takes to finally get it to where you've got a genome that can code for protein. That's got to happen. So the capsimers are built, new spikes, those are made out of protein, and then also we've got new genetic material. Once all of that's done, you've got the parts to assemble new viruses. They're put together, so you can see here's the icosahedral capsid with the genetic material. The last thing that has to happen is it pushes out to the edge of the cell, and it wraps itself in some membrane, so it gets enveloped here at this point. And that's, and for this particular virus, how it's released. So this is budding, which in essence is exocytosis. All right, the opposite of what we had up here. This is the reverse of that. So the virus leaves with a little bubble of membrane wrapped around it. Notice that the host cell membrane has those spikes incorporated in it at this particular juncture. All right, so an infected cell will start to show those spikes in its outer surface, indicating that it's about ready to start releasing virus. And this will continue for that host cell until so many viruses leave that it starts eating away at that cell's membrane, and eventually the cell will shut down, be incapable of metabolism because it can't afford to sustain serving that virus. Virus will wear it out. But for the virus's sake, it hopefully has produced enough of these infectious viral particles that it's releasing. It can go on and find new fresh host cells and keep going. If it can't do that, it's going to die out. So this adsorption step here is what determines which host cells can be infected, all right? And so some viruses have a real specific sort of set of cells that they can attach to and infect. So we say they have a very narrow host range. There's this term here, host range. If there are spikes, if there fibers, any molecule that they have on the outside with which they can attach to a host cell, if they're real specific for a particular set of receptors, the host range is going to be narrow. So, for example, some of the hepatitis viruses that infect humans, hepatitis B in particular, one of those hepatinoviruses, a DNA type of virus. Their spikes are so special and so specific that they can only bind to human liver cells. So if you had hepatitis B and you were shedding this virus, your dog, your guinea pig, your houseplants, other living things that you're around are not going to pick up that virus and be infected by it because it's that specific. Now, other humans, of course, could be infected by it if the virus finds its way to their liver cells. That's a really, really narrow, very specific range. Then you have some things that have a little broader range, like the polioviruses. The polioviruses can infect both intestinal and nerve cells, so they find receptors on those types of cells that their spikes will bind to. And basically all primates are susceptible to those polioviral particles. They're infection by them. So the primates include all of the New World monkeys, the great apes from the Old World, like orangutans and gorillas and chimpanzees, lemurs, tarsiers, all of those different kinds of things. Perhaps even tree shrews, I don't know. But it's like 110 species of organisms. Our close relatives, right? Forms that are in this order of primates. And so that's a little broader host range, right? And then we have forms like the rabies viruses, the rhabdoviruses, that can infect many mammals. So we know that all the carnivores, the skunks and the raccoons and dogs and even bats can carry rabies viruses. And if you're bitten by one of those animals that's infected, a rabbit, a wolverine or coyote or something, you could acquire the rabies virus. And so it's a broader host range, right? So sometimes viruses have a narrow host range, and they're specific for one particular organism, one particular host. Through mutation, they can expand their host range. And apparently this is what happened with the coronavirus. It's almost certain it came from bats. And it's interesting, I don't know exactly why this is, but bats tend to harbor viruses that can sort of jump from bats to other hosts. And of course, humans are one of those hosts. So it's an odd thing. I'm not exactly sure what would explain it, but mutations happen, and if the virus has the opportunity to jump to another host, it'll do it. And in the case of this coronavirus, it went from bats probably through an intermediate host, which would include pangolins or snakes. We're not sure which one of those it was. Pangolins are like a scaly, sort of anteater-like mammal. They're endangered, so they're selling them in markets. I don't know why that's allowed, but anyway, pangolins or snakes, and then from there they made their way to humans. So if you're in close contact for an extended period of time with those types of animals, they harbor a virus that could find their way into your body, and if the host range of that virus is broad enough, they could infect you. So that apparently is what happened. All right, so these images show viruses, and the image on the left here, you've got an envelope virus whose spikes now are bound to a receptor in the surface of a host cell. You can see where the receptors and the spikes meet up. They're shaped in such a way that they can attach, and there's obviously some other chemical kinds of activity going on there to help them to bind. So there's adsorption going on. And then this would be over here, examples of naked viruses that have capsid spikes. They likewise have found receptors which they can bind. That's what that looks like. And like I said, then once adsorption has happened, the genome has to come in and you have penetration and uncoding. Those typically are really sort of closely associated aspects of this whole cycle. Sometimes they happen simultaneously. But endocytosis, this is where you have the invagination of the host cell membrane, and it literally forms a bubble that wraps around the virus, which may already be enveloped. So the virus is taken and wrapped in that bubble of membrane, which then will have to be removed. Fusion is where we have an enveloped virus. So this fusion doesn't happen with naked viruses, but enveloped viruses can sidle up next to a host cell, and the envelope and the host cell's membrane literally sort of melt together, and the capsid comes in free of any sort of covering. And so then all that has to happen is destruction of the capsid to uncode it. And there's even other ways that things can go. So, but those are the two principal ways that penetration happens, is endocytosis or fusion. So here, these images show here at A. This is fusion, all right? This is fusion. What you're looking at here. So you've got the envelope, you got the host cell membrane, and you can see they sort of, there's the adsorption here with the spikes and the receptors binding. And here then you've got this merger of the envelope and the host cell's membrane. They're coming together, and here it is complete. So you see a little chunk of the envelope now jammed into the host cell membrane, and here's the capsid free of any envelope. So then uncoding takes place from it. This is good old endocytosis here. That's the endocytosis. All right, where we've got the fusion of, or the adsorption rather, of spikes and receptors, and the invagination step, the engulfment step, and you get your vesicle with the enclosed virus, and then ultimately the vesicle and the envelope and the capsid are all removed, and you get your free genetic material in here. It's DNA. And then this is endocytosis. This is also endocytosis. But what differs here is the uncoding. There's really not an uncoding. It's you've got your vesicle, but instead of that vesicle being destroyed, and instead of the capsid being destroyed entirely, the capsid's in here. There are apparently preformed enzymes inside of that virus that begin to bore a hole through the capsid and through the vesicle, and that allows then release of the RNA. So this whole assembly, the capsid and the vesicle, will remain behind. And if you have a virus that's like that, and enough viral particles were to infect the host cell, you're going to leave behind a whole bunch of these things. And those might be actually visible in the infected host cell for a time. So if you're, say, a pathologist, you're trying to determine what disease a person has, take some tissue and you biopsy that and you look at it under a microscope, you may see bunches of these little vesicles like this. Those would be indicative of a particular type of viral infection. I'm not sure what viruses do that, but there are some that do that. So you can see endocytosis can finish with uncoding that looks like this, or simply the release of the RNA without an uncoding step is the case here. All right, so there's a variety of ways to go. And these all show Well, they don't all show. This one here shows a capsid free of an envelope coming in. Anyway, this letter B here, where there's endocytosis of an envelope virus, well, that can happen with a naked virus too. It could happen the same exact way. Letter A here can only happen with an envelope virus. All right, so think this through. You know, what's going to happen with envelope versus naked viruses? What are the possibilities? Letter A can only happen with an envelope virus. Letter B can happen with enveloped or naked. And likewise here for C, although I think most of the time with C you're going to have naked viruses, but I can see where envelope viruses may do that same sort of thing as well. All right, so the next thing that we have to consider is, where does the activity involved in making new viral particles take place inside of the host cell? Once the virus comes in, if it's, say, a positive sense RNA virus, all of the activities involved in assembling new viral parts can happen in the cytoplasm. So that's going to happen fairly quickly. If it's a DNA virus, it turns out that the cellular machinery needed to process DNA is not in the cytoplasm, it's in the nucleus. So those viruses are going to have to go to the nucleus. So the classification system, focusing on the type of genome that the virus has, is significant in part because it highlights this aspect of the viruses, where they go in the host cell to do their work. So DNA viruses generally go to the nucleus, and it's there then that the DNA is going to be processed. It's going to be used to make messenger RNA, which then is translated into protein. There's a little bit of a lag there then between when the virus enters and when it has its effect. So it's going to take some time for that to happen. The RNA viruses generally do their work in the cytoplasm. And as is indicated down here, we've already mentioned those things. Positive sense RNAs can go immediately into translation, start producing protein and assembling quickly. Negative sense RNAs, those types of viruses are going to have to go through the step of making positive sense RNA, then they too can assemble parts. But that's going to happen in the cytoplasm. Then we got release. Like I said, budding is just simply the opposite of endocytosis. It is exocytosis. This picture over here shows some viruses being exocytosed. They're being budded. See, the viral particles now are wrapped with some membrane, which is color-coded red in this colorized image. And those are now infectious, and they're going to be equipped with spikes that allow them to bind the receptors of new host cells. So the thing about budding is the viruses that are released that way don't immediately kill their host cells. So it's sort of a protracted, extended period wherein the symptoms are present. But it doesn't lead to outright tissue damage and serious consequences right away. It takes a little while. But ultimately, that cell is not going to be able to keep up with the loss of membrane and loss of parts, and it will eventually succumb. Lysis is usually much more abrupt. Lysis is where you have a cell that has the assembly of lots of viruses, and they're all going to build up inside of that cytoplasm. You'll see their numbers building and building and building. The cell ultimately then will swell with all of those, and boom, it'll burst, and all of the virus is released explosively at that particular moment. So this little picture down here, this is actually a diagrammatic representation of budding. Lysis is where, let's say this is a bacterial cell, because these little X's are all the viruses. So as the viral particles build up and build up and build up, eventually that cell can't take it anymore, and it literally just sort of blows wide open, and you get a little, well, the outer covering goes away, and those viral particles are released all at once. Okay, so that's lysis. The operative word here is rupture. The cell literally release the viral particles. All right, so then we have this concept question. Viruses commonly contain DNA or RNA. Is that false? Is that true? It says DNA and RNA. Careful. Can a virus contain both at once? There is no. If we substituted the and in here for an or, if we plugged an or in there, that would make it true. All right, so what I'm going to do, I'm going to stop the recording here, because as I found, if I go too long, there are things that can happen with the internet, and it'll shut off on me, and I don't want for that to happen. So I'm going to stop it here, let you digest this one, and then I'll post another one shortly. All right. So keep it going.